First, we'll be talking about the digestive system. Starting off with the liver. This is a major organ that we didn't touch upon in lectures so far, but it is a very important part of our bodies. However, the majority of its many functions are not tested heavily on the MCAT. There are two functions that are tested decently amount, like a decent amount, and these are its detoxification function and glucose regulation functions. And we may have covered at least a little bit on its glucose regulation, but we'll be going more in depth with that, and as well its main function, detox. Okay. So detoxification. The liver is crucial in breaking down and eliminating toxins and toxicants from our body. We ingest either through our mouths or by breathing or through many other means, many toxins and toxicants into our body. And some of these we do accidentally, some of these we do like unknowingly and accidentally, others we do on purpose. Some of these include medications, like even this could be over-the-counter medications that we often take, or it could be more controlled substances and drugs and narcotics. Preservatives and other additives that are very often found in our food. Some of them need to be detoxed by our liver, right? And just because they need to be detoxified doesn't necessarily mean that they're like poisonous and they're going to kill you, but it means that our body doesn't like them and it can't break them down, so it needs to kind of eliminate them and treats them as kind of like toxins if they build up and stay there too long. Another very common toxin that we often consume on purpose is alcohol, and another one is caffeine. Okay, and there are many more, but these are just some common examples that you probably know in your everyday life. The first step to detoxing the liver is to neutralize or break down the toxins into more manageable intermediates. For example, alcohol is broken down into acetaldehyde and then further into acetate by certain dehydrogenase enzymes. These are called alcohol dehydrogenase and acetyl dehydrogenase, or ADH and ADLH, I believe it is. And these enzymes could be, like in this case, they could be dehydrogenases, or they could be some other enzymes that basically they just break things down from a very complex form that we can't really work with to something a little bit more manageable. Okay. However, oftentimes these intermediates are also very toxic and the body must get rid of them quickly, or there'll be a buildup and they'll cause damage to actually your cells in your body. One example that many of you may have seen is acetaldehyde, which is I mentioned is the intermediate when you're breaking down alcohol. It's very toxic in its own, and especially people of East Asian descent who have a poor functioning acetaldehyde dehydrogenase, which is the dehydrogenase which breaks down acetaldehyde into aldehyde. And people from East Asia usually genetically they don't have a very high functioning acetaldehyde dehydrogenase, and so because of that, when they drink alcohol, they begin to feel very strong symptoms of the toxicity of the acetaldehyde because it builds up because the body can't really do too much with it. And this is like the red flush, the nausea, the hot flashes, all of that stuff that you may associate with alcohol, and to a much higher degree because you can't break down the intermediate and you can't get rid of it quickly. Other toxic intermediates may also be free radicals, which can damage DNA and increase risk of cancer. This is the oxygen-free radicals, which you may remember from our DNA damage unit, or if you've learned about anything cancer-related before. Free radicals actually damage the DNA itself, and this heavily increases the risk of cancer. And our body has repair pathways, which we did talk about in our DNA repair lecture, has repair pathways that kind of try and fix these damages and neutralize these free radicals. But by introducing a lot of free radicals into our body through these toxins, that kind of overwhelms the DNA repair pathway, and there's more of a chance that there'll be a mistake, right? And that increases the risk of cancer. And all of this detox, it all happens in the liver. The liver is the main organ. And so that's the reason that for people who use a lot of drugs, who drink a lot, or who have a lot of chemicals or other substances that they put in their body, either through their food or their drink or anything like that, they can be at a high risk for things like cancer, but also different diseases of the liver, because these toxins can damage the cells of the liver. For example, for alcohol, one of the biggest risk factors for liver cirrhosis is alcohol consumption. So because the liver takes care of the detox, it is also the most at-risk organ if somebody does happen to take in a lot of toxins. The main things to note from this, at least first step of detoxification, is that the first step is always in the liver to neutralize and break down the toxins into more manageable intermediates. But once they're in the intermediates, you still want to get rid of them, because the intermediates are often pretty toxic themselves as well. And if you ever see something like this on MCAT, they'll tell you there's this substance that in your liver gets broken down into this intermediate or something like that, and you just have to answer questions based on that. They'll give you the example, so you don't really have to memorize like alcohol gets broken down into acetaldehyde and by what kind of enzyme, what's the name of the dehydrogenase or something like that. Okay, so let's continue on. Once the first step has been completed in the liver and the toxins have been reduced to intermediates, a process known as conjugation, using amino acids or amino acid derivatives such as glycine, glutamine, or taurine, which is not an amino acid, but it is sort of like an amino acid, so like a derivative, converts them into easily eliminable end products, which can be sent to the kidneys or to the bile, which will lead to removal via urine or feces, respectively. Right? The only two ways we can excrete something out of our body, actually, there's three ways. It's either back out through your mouth, either through breathing or by vomiting, which usually we try not to do, or through urine or feces. The other two ways, and that's usually what we do. So you break these down into something that's very easily eliminable. So for example, like sometimes carbon dioxide, water, other sugar molecules, whatever. And then you just send them to the kidneys, and then from the kidneys, those chemicals will be kind of washed out and filtered out, and they'll go through the bladder and out the urine, or you'll send them to the bile, which will lead them to get rid of through the rectum, right, and in the stool. Okay, so fairly simple, and we're not going to go into for the MCAT purposes, it's fairly simple, but what you may have to be aware of is that they may give you a mechanism, like for example, the one I gave you with alcohol, they may give you a mechanism for another chemical or some medication or something like that, and you have to recognize how it's being broken down, and that requires a lot more biochemistry knowledge rather than necessarily knowledge of the liver functions, right? So be on the lookout for that if that ever shows up. Next up is the liver and its glucose regulation. So the liver is a very large organ, and one of its main functions is the storage of glucose as needed. Using the signal molecules like insulin and glucagon, the liver will either take in or release glucose to and from the blood, right? When we talk about sugar, we're usually talking about our blood sugar, and that's usually what you measure. If you have diabetes, or how you know you have diabetes is the blood glucose level. As you remember, glucose is stored as glycogen in the body. When you're not using it, it's stored as glycogen. And the glycogen is actually stored in the liver. That's the site where glycogenolysis and gluconeogenesis occur, where you take the glucose to the liver, and in the liver it gets turned into glycogen, and then the glycogen, when needed, gets turned back into glucose in the liver and then sent back out to the blood where it needs to go, right? The insulin glucagon receptors on the liver are very crucial to regulating the blood, because that's the site of gluconeogenesis and glycogenolysis I mentioned, right? Again, a lot of this, just so you know, it does happen in the liver, and the liver kind of, if it gets damaged, it may, like if the sensors in the liver get damaged, it may very heavily impact the rates of gluconeogenesis and glycogenolysis. They ask you that, something like that. But for this, mainly, you do have to remember, what is gluconeogenesis? What is glycogenolysis? What are the steps? What are the conditions? Things like that, right? But no, again, this happens in the liver. The liver gets damaged, and the receptors, especially if the receptors for insulin and glucagon on the liver get damaged, that could lead to heavy problems in glucose management. Either you won't have enough blood sugar, or you'll have too much blood sugar and be diabetic. Okay, now that we talked about the liver, and again, the liver has many functions, many like more detailed functions, but we're just covering the main ones. Okay. All right, moving on to the large intestine. And while we think of our large intestine as just an organ, it's actually a sprawling ecosystem for many different types of bacteria. It's estimated that about 300 to 1,000 different species of bacteria can live in our large intestine. More commonly, there's about 50 different species that make up like 90%. 90% of our bacteria in our large intestine are within 50 species, but there can be as many as 300 to 1,000 different species living in our large intestine. So it's essentially a whole ecosystem down there, right? The vast majority of these bacteria are anaerobes, meaning that they do not need oxygen to make energy. They just need glucose and other molecules. They don't use oxygen, okay? That's different from our cells, which are aerobes, right? These bacteria have a symbiotic or mutually beneficial relationship with us. Remember, symbiotic means it's good for both parties. They greatly help both our digestive and immune systems. And in exchange, they get to live in our stomach and they have an ecosystem there, and they sometimes eat scraps of our food. Okay. So what are the benefits of bacteria? Usually when we think of bacteria, we usually think of bad things, and we don't want any bacteria in our bodies. That's how we get sick. But in your mouth, in your large intestine, everywhere, you have a lot of bacteria, and a lot of these bacteria are actually very good for you, right? Firstly, these bacteria greatly help the immune system and overall protection of our body from pathogens. Pathogens are like bacteria and viruses and things like that that will cause diseases in us. These bacteria that live in our bodies are not pathogens. They don't cause disease. They just live within us. And because of that, they cover nearly the entirety of our habitable large intestine. And thus, they kind of outcompete any foreign bacteria that we could ingest, right? If, let's say, we have our large intestine like here, and all of this is taken up by our good bacteria, which live in our intestine, any foreign, do it in red, any foreign bacteria that come in will have to take up one of these spots. These spots essentially are all already taken by our good bacteria, and our good bacteria will kind of like not let them stick on us, and essentially they won't let them take hold. And once they take hold, they'll start multiplying and they'll start doing whatever they want, and that's how we get sick. But our bacteria kind of take up all the space and they prevent that from happening. In addition, our gut bacteria may also release cytokines, just like our immune system does. Like our killer cells also release cytokines to kill bacteria and viruses when they see them. Gut bacteria also release cytokines. Some gut bacteria release cytokines to actively combat and kill the foreign pathogens, because obviously it's in their best interest not to lose the fight to these pathogens either, because if they lose, they will give up their spot, and the bacterial and the foreign invaders will take over. So they want to keep their ecosystem to themselves, and so they will protect it, right? And that's good for us too, because we also don't want these foreign invaders living in our stomachs. Another great benefit of the gut flora is their aid in our digestion. There are certain polysaccharides, starches, sugars that we eat that our bodies can't break down. But the enzymes provided by these gut bacteria can break them down. And once they break them down, we can actually use them and get energy from them as well, right? Without these bacteria ingesting these molecules, would lead to low benefit. Without these bacteria ingesting these molecules would lead to low benefit for us. Thus, we would need to consume a lot more food in order to receive the same caloric benefit out of it, right? If you eat a bunch of like wood off like a tree, you can eat and eat and eat and eat, and you won't really get much benefit out of it because you can't digest the wood. right? And just like you can't digest like rocks or something like that. It'll go in and go right out. Same thing with these polysaccharides and these sugars that our bodies normally can't digest. If you couldn't digest them and you just ate them, they'd go in and then they'd just go right back out and you wouldn't get any benefit from it. But because these gut bacteria live within us, they kind of break them down a little bit, and then we kind of take a little bit for ourselves, and we can use something out of something that we couldn't have before. There have been studies that shown if you completely get rid of these gut bacteria in other mammals, like in rats or whatever, that they will have to eat many, many more calories per day in order to keep the same weight, in order to prevent themselves from losing weight than the same exact mice or rats if they just had these bacteria. Okay. Going into a little bit of medicine, these benefits are one of the main reasons why it's advised for medical professionals to not overprescribe antibiotics. So antibiotics kill bacteria, and it may seem like, oh, what's the harm if you're just killing bacteria? Well, firstly, one of the main reasons that they say not to do that is because the bacteria will grow, as a species, will grow resistant to the antibiotics, and over time, our antibiotics will stop working. But another big reason is that you actually need bacteria in your stomach to, like, you can see all of these benefits. You actually need these bacteria; they're actually helpful for you. So if you just keep constantly taking antibiotics, the antibiotics don't discriminate between bacteria, so they'll kill everything. And if you take in too many of those when you don't need it and it's unnecessary, they'll kind of eliminate many of the gut bacteria as well. Similarly, this is why it is also healthy to consume probiotics, right? There's antibiotics and there's probiotics. Probiotics are things like yogurt and other like dairy products that have, or sometimes now we have like just special like probiotic things which have just a mesh of bacteria in them. But natural probiotics are like yogurt and stuff. And these probiotics contain a lot of these healthy bacteria, especially now with more modern science. We can actually control and determine which exactly bacteria species we want in there. So these probiotics replenish your gut flora and make sure you have a healthy amount of those, right? Next, let's talk about the muscle control of the GI tract. So our GI tract has a very complex system of neurons controlling it called the enteric nervous system, or ENS. Many consider this kind of like a second brain, as it's independent of the brain itself and the spinal cord. Although it can be influenced by them via the parasympathetic and sympathetic nervous system, parasympathetic nervous system is more like the rest and digest. This can influence the stomach to, okay, start digesting stuff now. We're resting, we digest. Or the sympathetic nervous system can tell them this is not the time for digesting. We need to fight or flight or something like that. So they can be influenced by the actual main nervous system, but even if you cut off all, like, and there have been experiments where on animals they've completely cut out the connection between the main nervous system and the enteric nervous system, and the enteric nervous system still continues to function, although at a reduced capacity, of course, because again, you do need the parasympathetic and sympathetic nervous system, but still it continues to function even without any outside interference at all. That's why people call it like a second brain. One of the main jobs of the ENS is to control peristalsis, which is the muscular contraction of the GI tract used to move food along it, right? So you need to move food from like your mouth down to your stomach, and you need to move it past. So how do you do that? Well, you just take the ball of food and then it's going down the tube, which is your esophagus and then your GI tract. Essentially, you'll just squeeze it, then squeeze here, and squeeze here, then keep going. And the food, when you squeeze it, it'll move to the right, to the right, to the right, to the right. It'll essentially like, kind of like when you squeeze like toothpaste out of a tube, it'll go right out along your GI tract, right? And this squeezing is called peristalsis. And peristalsis is essentially completely controlled by the ENS. And what that means is You know how when you have your brain going to your skeletal muscle, it starts in your brain, then it goes down the long axon to the end plate, to the axon end, and then there's the acetylcholine going to the muscle, and then it excites the muscle, and the muscle begins contraction. Well, this all happens exactly the same way, except it doesn't start at the brain. It starts in this complex web of neurons called the ENS. So it starts in your stomach, and then the stomach releases the shock down the axons or wherever, the equivalent of the axons, to the GI tract muscles. Again, go to the motor end plate. Acetylcholine goes to the muscles contract. Peristalsis occurs. Okay, so peristalsis can happen completely independently of the main nervous system. Without peristalsis, the food bola, which is like a ball of food, right? When you chew it and swallow it, it turns into a ball, like a mush, and it's called the bola. Without peristalsis, the food bola would never move through the esophagus or the intestines, and the full muscular control process is regulated solely by the ENS. Okay. All right. So the ENS is also crucial for controlling the secretion of necessary hormones such as secretin, which signals for the release of pancreatic and stomach enzymes needed for digestion. We talked a lot about those in the main digestive system lecture. All of those are released by secretin, and what determines whether or not secretin is released is ENS.